Okay, class. So here we are back, and in today's lecture, we entirely have to see that how come the ultrastructure that we have got for the plant cell, this is gonna look like. Earlier, I have cleared it in the previous class. In that sense, the eukaryotic organisms are concerned, so they have got the membrane-bound nucleus in them, and the plant cells, they are the eukaryotic cells, and we will see that they have got the nucleus in them. Now, before I show you the diagram of the plant cell, now here you must notice as that whenever we have the plant cell to be discussed, so what structures do we have to be identifying in case of the plant cell? So in these structures, you may write the name somewhere that initially what we will have in the plant cell, there would be the presence of the nucleus, and the nucleus has got further subdivisions into it. And as these subdivisions are concerned, so the nucleus would have got its own membrane. And the name of the membrane, it would be entitled as the nuclear membrane. Then within the nucleus, we have got another component coming. It would be the nucleolus. And then we have got the third thing in the nucleus, and they would be the pores of the nucleus. And we call them as the nuclear pores. The thing doesn't end here. In the nucleus, we have got something else like usually the cells they have the cytoplasm in between the nucleus and the cell membrane, but for the nucleus there would be the nucleoplasm. Moving onward, in a cell when this is not dividing, so we have got the genetic material, and that genetic material is in the loosely arranged form, and we refer to this as the chromatin. Moving further from the nucleus, we will see that in the plant cell, there would be coming up of the membranes, and they are the rough endoplasmic reticulums. And then we have got something else that goes with the name of not RER, but rather than we call it as SCR, and the SCR stands for the smooth endoplasmic reticulum. Moving onward, we have got for these plant cells the third thing coming, and you know it from your previous classes that this would be the large vacuole with us. And in the large vacuole, we further have got its membrane, and the membrane of the large vacuole would be entitled as the tonoplast, and there would be the solution filter within the plant cell, and that we regard this as the cell sap. Moving onward, now this is some of the misconception that we believe that the plant they make their own food, so they would never require the mitochondria, but this is not the case. The plants they will have the mitochondria in them, but the difference is that when we give the nutrition or we give the nutrients, the organic nutrients to the mitochondria of the animal cells, so they come from the food that we eat. But since these plants they have got the chloroplasts in them, okay, and these chloroplasts, you see, they would be making the glucose, and that glucose would be carried to the mitochondria where they would be doing the respiration. The thing doesn't end here. For the plant cell, the sixth component that we have got with us, so that would be referred to as the Golgi apparatus, we say. The word is Golgi apparatus. Now, this is an entirely new term that we don't encounter in our junior classes. And in the Golgi apparatus, further, we have got two of the things. Among them, we have this Golgi body, and along with the Golgi body, we have got these vesicles. And these vesicles, they are the Golgi vesicles with us. So the Golgi body and the Golgi vesicles collectively they make the Golgi apparatus of a plant cell. Moving further, in these plant cells, we have got some other spherical structures, and these spherical structures, they are the ribosomes with us. The ribosomes can be attached to the membrane of the rough endoplasmic reticulum, or they could be dispersed freely within the cytoplasm. And how can we forget about the other basic things that we have got in these plant cells? So you need to remember their names as well. That here we have got something that would be present in the plant cell, and this would be the cell membrane with us. And then who doesn't know that the plants they have got another component in them, and that would be the cell wall. Now there would come another, you could say, new term that you may never have encountered before, and that are the cytoplasmic kind of an interconnection between the cell walls. And these we refer to them as the plasmodesmata. And unlike the animal cells, so these plant cells they do not have any of the centrioles in them. So now these are overall structures that we have got in case of the plant cell. And now here I'm gonna take you to each and every component. So overall, you see that this is the plant cell with us. So this is one individual plant cell, and this is the neighboring plant cell that we have. Okay. Now I would start from the very, you could say, outside. So you see that here what we have got on the outside. Now this would be the cell wall. And when I'm going to come downward the cell wall, so here you see the component that I'm highlighting with the blue marking. So this is the cell membrane. Now, moving further, if you were to see over here, so here we have got this important organelle, and this important organelle overall, if I were to be highlighting it with the laser pointer, so this is the nucleus, and the nucleus has got on outside of it, that would be the kind of a thing that we must be calling this as the nuclear envelope. Okay, so we have got this nuclear envelope for the nucleus, and if I just zoom in, so here you see for yourself that we have got these pores, and these are the nuclear pores. And at the center or within the nucleoplasm of the nucleus, we have got this nucleolus, and then there would be the genetic material that when the cell is not dividing, as I mentioned earlier, so these are the chromatins. And in close association with the nucleus, so here what we have got now, these are the membrane of the rough endoplasmic reticulum, and the rough endoplasmic reticulum usually they lie in close association with the nuclear outer envelope. One thing that I have said earlier when I was writing the components, so here we have got the protoplasmic mass within the nucleus. Like here we have the cytoplasm, but within the nucleus we have this nucleoplasm. I have highlighted that this is a rough endoplasmic reticulum. On the rough endoplasmic reticulum, here would be the small spherical units being present, and these are the ribosomes. And this is the presence of the ribosomes that makes the appearance of these membrane as rough, so that is why they are entitled as a rough endoplasmic reticulum. Now, moving further, we don't have any centrioles in them, but here we have got another important organelle. Now, this is the mitochondria. This has an outer membrane, and also these mitochondria, they have got an inner membrane as well. If I were to take you further, so at this point, you see that we have got these chloroplasts. And these chloroplasts, how you have to highlight them in the paper, that there would be presence of the structures that would look like this. Okay, so this is just simply the identification that you need to know for the chloroplasts. If we were to contrast between them, so the mitochondria have got this inner enfolded membrane, and here within the chloroplast we have got the structures like this. These are the membranes that we will learn later that these are the thylakoid membranes, so they appear like this. So this organelle is the chloroplast. And again, here again we have got the mitochondria. Now, if I were to contrast these membranes that are lying over here to the rough endoplasmic reticulum, so they do not have any ribosomes onto them. So these membranes that we have here that I'm highlighting, so these are the smooth endoplasmic reticulum with us. Ribosomes, they were attached to the membrane of the rough endoplasmic reticulum, but the ribosomes, they can also lie freely dispersed in the cytoplasm. Now, if you were to come here, so you see that here we have got at the center of the plant cell is this large vacuole. And the usual thing or the points that we must know for this large vacuole is that at the center there would be this large vacuole, and the large vacuole has got its own membrane. And the name of the membrane for the large vacuole, this is entitled as a tonoplast. And then there would be the filling up of the, you could say, solution or the fluid within the large vacuole. And this you have to be remembering that we entitle this as the cell sap. So this is the tonoplast of the large vacuole. And here the fluid filled space, all of it that we have in the large vacuole. So this is cell sap. Then I mentioned that there is something that comprises of two things, like we will have these Golgi bodies, and these Golgi bodies, along with their Golgi vesicles that we have here, so they would collectively make the Golgi apparatus. Then I mentioned something that the adjoining plant cells, they are connected, or their cytoplasm, like this is the cytoplasm of one plant cell, this is the cytoplasm of another plant cell, and you see that these are their cell walls. And in between the cell walls, we have got these perforations here, and these perforations, they connect the cytoplasm of one plant cell to the adjoining plant cell, so they are entitled as plasmodesmata. Okay, and then here the point where the two cell wall they're gonna attach with one another. So we will have something coming here, and this would be entitled as the middle lamellary. So the middle lamellary is lying here. So this was all about the ultrastructure that you must know that how come the plant cell this is gonna look like.